Thank you again for inviting me to share a little bit about what we're going to be kind of doing to support the biomed research and LBRN research project. So I want to briefly talk today. What I've been asked to talk about today is primarily about the way that we can support different kinds of research projects that are being run through the biomed sequencing core, and what types of questions and answers are possible through that, and also how can you kind of leverage different resources that are kind of built into that cost structure that is available through biomed. So first of all, I think what we discovered is that a lot of people that potentially could be doing the sequencing struggle not only with the standard processing tools, but also then having someone to talk to and really going deeper into their data. And so we've developed this process of first gathering the requirements and then providing some standard processing outputs that are very predictable in terms of what it is and how much it costs and how long it takes. And then being able to work with the researchers to answer their custom research questions and also provide them with the support that they need to insert any kind of figures in their publications or anything like that. So I don't know exactly what is the right link for GeneLab. But maybe Ramesh or someone can post that, because I think that's where you would be able to get all of the sequencing capabilities. And I think what we provide on the analysis side is really tied into whatever sequencing they offer there.
GeneLab services are available through iLab. You need to get an iLab account, and then in there we have the services listed. But we can talk about that after your presentation.
And so to showcase how this works in practice, I'll talk about 3 projects. I'm not going to go into the details. I think most of the projects are maybe at different stages of being published, so I'm not going to go into what the biological question was or what the design was. I'll just show kind of some of these stages that I'm discussing here and what are the outcomes, what do you get at the end. So I think one of the most common projects, RNA sequencing, typically one of the limitations of an RNA-seq process project is that you get your standard outputs, you have the tables, but then a lot of questions only start appearing after you have that initial processing done. And so our goal was to really develop a standardized pipeline. From that pipeline, provide all of the different outputs that could be potentially interesting, from gene to isoform expression to annotation, and kind of then be able to work with the researcher to understand what are the specific questions and provide them support from statistical analysis and kind of exploratory analysis all the way down to pathway annotation and supporting their specific research questions. And so here you can see right away that, the time that it typically takes us, so the sequencing core would sequence the samples, and then that would be uploaded to the Illumina Basebase account. And from there, we take that, and very quickly we can produce the initial tables for you to kind of work with if you know what you're doing, or if you have some additional requirements like differential gene expression, downstream annotation, then that can take a little bit longer, because typically then you would want to focus on some specific element and get it down to a very detailed view of the biological questions that you were trying to address. Another example project that I have here is something that is a little bit more custom and not as widely used, I would say. So TCR sequencing. These are T cell receptors, CDR3 receptors, where not only is there a limitation, I think, in what the standard output is, which really just is a proportion and quantification of clonotypes, but also a lot of kind of results that are difficult to deal with. For example, in this project, the end result was a combination of kind of novel sequences that no one really knew what to do with. And so all of a sudden, a lot of questions appeared. What do you do with all of these sequences? Which ones are the more important and significant for the treatment that was being studied? And then also, how can we kind of create some kind of large-scale process so that we don't have to deal with this in a manual way, going through BLAST and kind of annotating results manually. So what we were able to do here is to use statistical analysis to identify clusters of clonotypes that were really more abundant in the specific types of treatments that were being researched here, and then use some standard tools to predict epitopes with good binding properties, and then use a distance matrix or a dissimilarity matrix between the sequences of these predicted epitopes to then start asking questions about what are some of the expected and unexpected epitopes that we're finding to really try to annotate also a lot of the unknown sequences that were identified. And so this project took a longer time, but I think it's a good example when, you know, someone has maybe some specific questions in mind, and it's typically a bioinformatics, you know, kind of a challenge to get stuck at a certain stage and then try to identify some, I think, creative ways to get out of that situation. Finally, another example is an example with single cell sequencing, where I think 10x genomics is a new a new capability that the center has now. And we were dealing with a project where different time points for different cell types were really, you know, a lot of information is there, but we were tasked with just kind of annotating the different types of cells, and then using a standard procedure more or less today, but to really study how the cells differentiate over time, or the pseudo time trajectory. So we were able to not only, I think, kind of work through the process of eliminating some of the noisy data that came with the project, but also perform all of these tasks in a very short period of time, and then start kind of asking those specific questions about, you know, what are the marker genes and how do these marker genes behave across time and between different cell populations. So those are some of the examples, but I want to summarize this part of the presentation to really talk about some of the reasons why this makes sense. So first of all, we work closely with the team at the Biomed lab to really understand you know, all of the information about the samples that they have, about the preparation, about the protocols that they use. And that allows us to fine tune our standard processing to the way that they process those samples on their end. I think we've also established a fairly efficient process of passing information and the data between the kind of biological research side and the bioinformatics side to be able to support the researchers that have questions that kind of span that whole process. You know, they can send out the samples, or maybe they have questions about experimental design. And so I think that is really a major part of making the bioinformatics process effective. Another one is standard and predictable pricing. So I think it's very important for researchers to be able to include that pricing in their proposals, to plan their experiments, also for LBRN to be able to support those projects. And so I think there we have a lot of flexibility with how little or how much we're able to take on at a specific project. And another important part of this, I think, is that we have a close collaboration with the Tauber Bioinformatics Research Center. The center develops novel algorithms and is working with a lot of collaborations where new algorithms are being developed to address challenges that, you know, don't have really effective solutions or really need something new to be developed. And so the benefit of that is not only that they develop these algorithms, but we're able to package those and make them a part of a standardized environment where it's very easy and you know, possible to use them in a user-friendly way. And so to transition to that, I want to briefly mention we have a platform that is actually designed for that use case. So as a support service, oftentimes these kinds of analyses can be time consuming and they can be very difficult to predict. You can have new questions appear as a result of different stages of the analysis. And oftentimes it's not economical to try and have somebody service every step of the way. So the platform allows a biologist to be much more independent with these complex tasks, mostly for processing standard, getting standard outputs. But I will show you today a few examples of how you can also go into the exploratory stages and also in downstream annotation. So as I mentioned, single cell has been an emerging technology. A lot of people are starting to use it more and more. And so we developed a dedicated section that now allows you to try out different pre-processing methods, different batch correction methods, try out different types of visual outputs, and all of that in a point and click interface that really makes it easy to get all of the standard outputs that you might expect in a single cell study. Not only that, we also provide training modules if you have some data set that you would like to continuously work on, and you have a student, a graduate student that would like to kind of get involved into that project. We provide the full support from that kind of standardized package pipeline to Colab notebooks that you can go through and kind of modify parameters yourself, and also develop that into a reproducible workflow that later on you can use on your own. In addition to that, with a lot of sequencing being done for SARS-CoV-2, we developed a number of tools and training resources that you can use to plan those kinds of experiments. And one of these, I think, focuses not only on kind of standard consensus annotation, but also the ability to develop these intermediate files where you can start looking at subconsensus analysis. We also worked with the LBRN core to help annotate and process a lot of the samples where the major question was annotation of variants of concern. And there we developed kind of a full pipeline that automates identifying specific positions, annotating them based on the known kind of variants and their classification, but also being able to generate intermediate reports to ask deeper questions. One of those types of questions is really around how does the sequence affect the structure? And so we kind of put together that first initial stage of identifying the variants and then also producing variant mapping onto the structure of, let's say, the spike protein or known structures to help kind of investigate and study some of those questions at a deeper level. And finally, because I took over after Chris, he's really an expert in metagenomics and does a lot of that kind of work. We try to simplify and make a user-friendly version of working with metagenomic data. And these are just two pipelines that we have, data2 and KIME2, where you can not only process the data and learn about those kinds of analysis methods, but also at the end get some interactive visualizations that you can customize and on your own incorporate into your research. So these are just some tools that I think supplement what we do on the service side. After the sequencing is done, from initially setting that expectation to managing the process all the way to supporting the publication stage, and also giving tools and support services to continue and study that data, because as data accumulates, new questions really appear. And so far, we've been able to work with a number of institutions in the state to support their research projects. And so if you have any questions about that or the standard pricing, Ramesh will be able to share that information with you after that. Now, briefly, I also want to mention another part of what we do, and I think Chris already alluded to that. A major challenge is not only in having the knowledge of bioinformatics and even having practical tools, but also following a proven process of learning that allows students from different backgrounds to acquire skills and become independent in their approach to biological questions using data. And so we've developed our OmicsLogic training around that challenge to really support going from the basics of terminology and understanding to an understanding of how experts use some of these tools in their research projects, and then becoming independent and being able to apply those to research questions of their own. And just this January, we launched a pilot with the National Association of Idea Principal Investigators, where students from several different programs from around the country participated in a discussion and also a pilot program to make these courses available throughout the network. So kind of from Louisiana, exported to a lot of these new places. And so we're excited to say that we had over 100 participants, both faculty, undergraduate, and graduate students. They were really successful at going through the process. And so for you to appreciate what that process looks like, I have just a couple more slides to explain what is it that we offer and how is it different from the traditional workshop approach or maybe coursework in traditional programs. So first of all, we have a process that allows both coding and non-coding paths to learning and mastering bioinformatics. And what that means is that you can appreciate bioinformatics, but maybe not have enough time to master all of the different requirements for the technical all the way to statistical and machine learning. And so there's really a lot to learn. So our focus is to making it available and lowering the barrier of entry. And we do that by packaging these complex technical things into short coursework that is available online and can be taken asynchronously. And so from this pilot program, we were surprised to see that a lot of the participants, a lot of them were from Hawaii, from UNMC in Nebraska, from other places, were able to go through that whole process. And we had these 5 options that people could choose from. Introduction to Bioinformatics, which is a very basic program, introductory program for someone who doesn't know anything about it. Data Science, which is focused on machine learning, statistical analysis, coding in R and Python. Genomics, which is focused on variant analysis for cancer and infectious diseases, metagenomics, and bioinformatics for infectious diseases, primarily because there's such an interest in COVID-19. And so what that means, and I'll just give you 2 examples here. In our Introduction to Bioinformatics program, which is, by the way, available for free across NEPI and LBRN, is a brief introduction to the applications of bioinformatics, the need of bioinformatics in different domains, the intersection between biological and data concepts, and an introduction to the technical side of bioinformatics, which is coding in R and Python. In addition to that, we have specialization tracks that are really papers from that domain, explained in terms of their bioinformatics methodology, providing with the technical and reproducible tools for someone else to replicate that study. And that it covers oncology, infectious diseases, neuroscience, and agriculture. Now, many of these examples are, like I said, available through coding and non-coding tracks. And that means that we have a variety of tools that one can use to produce plots that are standard and used, and learn along the way while building pipelines what those pipelines represent and how do they transform data into something that is biologically interpretable. In addition to that, for those that decide to go deeper and beyond that, we also offer a full process from context to the logic of the analysis to then practical tools that they can kind of learn themselves using different tools like Google Colab to not only do the analysis, but then also save that code that they could potentially replicate in the future. And so, like I said, one of the major objectives is to elevate the level of biomedical research by bringing a lot more students into the bioinformatics space and giving them the tools to get started quickly, because I think most of the learning actually happens when you do the learning, when you do the analysis. And so as you struggle to come up with the results and you have to find out how to do a certain figure, that's really when you learn. So that's really the concept of that. And to support that, the online portal that we have designed called OmicsLogic And I'm just going to put the link here in the main chat. If anyone wants to take a look at that, it is learn.omicslogic.com. The sign up is free. Anyone can get started. There are a lot of free resources available. Moreover, it's very easy to go from the very basic and free to the much more advanced and kind of research-oriented types of activity. And those include not only activities for learning, but also activities to understand how students learn. And that is another important topic, I think, in the domain of bioinformatics and data science education, is that the approach is to give tools and kind of show skills. But in reality, the learning happens with a lot of variation across different types of learning styles and learning techniques. And so we're always excited to incorporate new ideas that come from experts that are already a part of this exciting network. And so just as an example of that, we've been working with LBRN to identify important resources that are already being taught and used throughout the network, to incorporate them into our learning portal and make them available to students across the network. One of these is by Dr. Himanshu Raje from Nichols State University, a graduate of LSU, who developed a course that is important for students who are joining any kind of biological curricula, to incorporate the data component into that as well. And so he goes from the very basics of understanding what does biological data look like, to a little bit of coding and understanding how to use packages in Python, so that one is able to appreciate the need and the application of bioinformatics in life sciences in general. So to support that process, we're actually making all of these resources available for those who are interested to contribute coursework and make that coursework available throughout the network. And that means that we can support development of training materials, develop easy to use data analysis tools, help analyze their data set and turn publications into a tutorial, and also help develop interactive data stories around those data sets and important research topics, so that students can really benefit from that. Now, I'll finish with my last slide, where I'm gonna just mention some upcoming opportunities. So recently we started this discussion about this upcoming summer. What is really important in the summer research program? How can students get some training and expertise in bioinformatics? For the previous 2 summers, I think we had a lot of interest. Between 50 and 100 students each summer were participating in the research training on different topics. And we often have a lot of different topics at the same time. So I think on one hand, that's a benefit. A lot of students can pursue what they are already interested in. In another way, that's a limitation, because people don't understand what goes into that training. And so what we're going to do this summer is we're going to talk about single cell and bulk RNA-seq transcriptomics and cancer. We're going to talk about COVID-19 again, and continue offering training on variants of concern, as well as microbiome and metagenomics in long COVID. We'll have a training program for machine learning and data visualization in bioinformatics using Python and Plotly, and building interactive dashboards to make exploration of data easier. And we hope to introduce a new specialization, which is going to be focused on agrogenomics. And we'll use the example of cannabis to talk about genomics and transcriptomics for agriculture and biotechnology. So with that, again, thank you all for listening to my presentation. I'm happy to answer any questions that you might have. And if you're interested, you can also get in touch with me directly. My email is elia@pine.bio. Thank you, everyone.
All right, if anybody has any questions for Chris or Elia, the floor is open. That was very informative. By the way, Chris is the CEO of Pine Bio. I missed introducing him. And
Elia, not Chris.
Yeah, it's my apologies. Once again. Elia is the CEO of Pine Biotech, and you mentioned the Tauber Institute, and I think they've had located at University of Haifa, and the bioinformatics part of it was led by his dad, Dr. Leonid Brodsky, and he's been a tremendous asset for us. And many of our researchers in the LBRN network have availed the services of Pine Biotech through the LBRN core, through the Gene Lab core, and I'd like to believe that so far they've been happy, and I've gotten feedback from a bunch of them saying they are very happy with the way interactions have taken place. Anybody has any questions for Dr. Christopher Taylor or Mr. Elia Brodsky? Okay, all right. Gus wants to say something. We'll take, now is the time for a brief pause. We are actually 5-7 minutes into our pause. So we'll reconvene in 7 minutes time for our next session. And Gus will introduce our next speaker then.